as I sat down, I have to confess that my colleague Dario asked me to come and give this talk in his place, and the title of the talk had already been defined as the paper of the future. And so when I sat down to put slides behind the title of the talk, I've gone through some of this in my history.
No.
So I have a little bit of PTSD from this conversation. So I spent the better part of my year, let me back up and say, I sort of come to this equation having been a former academic librarian for many years at the University of Washington. And then I went into the tech industry, and for the bulk of my time at Microsoft, I was working for an e-science lab within Microsoft Research. And part of what we were doing was looking at some of the data-intensive approaches to science, and then sort of as a reflection of that, how does science communication happen in a data-intensive world? What are the artifacts that we're creating? Software, data sets, how can we enable reproducibility? And I dug out from my slides this slide that dates from 2005. This is a Microsoft slide from 2005 that talks about the new era of research reporting, where it enables reproducible research. You have interactive data, you have collaboration, some dynamic documents, and then just reflect on the sort of Jupyter Notebook talk that we just had. The person who gave this talk was a guy named Bill Gates at a conference called Supercomputing in 2005. And Microsoft, being a company that made proprietary software at the time, not always loved by the academic community. We were trying to sort of find some ways that we could build on top of some of the platforms that we had at Microsoft. So I was involved in building some free open source tools out of Microsoft Research that extended some of the core functionality of Excel, of Word, of some of our other things. But just to give a couple of examples here in the Word space, we built something with Phil Bourne, as a matter of fact, and Science Commons at the time, called the ontology add-in for Word. And the general problem statement was that people are writing their research reports and submitting them to publishers that get then enshrined as a PDF, and you're now dealing in the world of natural language processing, and terms are very ambiguous. So what would happen if we took some of the biomedical ontologies and we grafted them inside of Word? So Word now becomes a browser that can communicate over the web to the UMLS ontologies and can light up your document anytime a word occurs that has a correlate within one of these ontologies. And the author of the document then is in control of saying, yes, that string right there is a protein ID from the protein data bank, or that term right there is this drug that I'm referring to in this context. And all that data would be, semantic information would be encapsulated along with the Word document that the publisher could then unpack. So rather than using NLP processes on a PDF, you sort of move the problem of identification upstream. It's a great idea. Nobody ever used it. I also worked on a project with Peter Murray Rust out of Cambridge University, which again, the problem statement was in the chemistry domain, you have a bunch of different ways of representing chemical elements, chemical concepts in a paper. It can be a chemical formula, it can be a name, it can be a two-dimensional structure, it can be a three-dimensional structure. And the chemist can look at that and decide what that means. But if you're trying to mine that information, it becomes very difficult. So we built a chemistry authoring environment, again, open source, embedded inside of Word, that allowed you to put structures and strings into the document, but to have this backed with an XML chemical markup language representation of the molecule. People did use this. This is still an active open source project right now. It has a couple thousand downloads a month, but publishers aren't using it. Part of our goal was to have this semantic information flow through the pipeline so that by the time it shows up as a published chemistry paper, you have something that is queryable and searchable, and that part never really materialized. And then we did some work with the Broad Institute for reproducible workflows and execution of reproducible workflows from within the Word client. So you could actually define a workflow that was going and searching the gene pattern database, munging the data, and then building a visualization out of it, sort of like a Jupyter Notebook could do today, and then insert the updated version of that image into the document. So the workflow, the actual query parameters, the data set all sort of travels along as part of this Word package, again, to be consumed that never happened. In addition to sort of building things at Microsoft Research, we were also looking at some of the landscape for quickly sharing scientific information. So just to pick one example that Michael Keller referred to earlier is archive.org. And we worked with and funded them a lot of the protocol work as well as some of their early projects there. So when I say I have PTSD, we've been talking about, not just me, but the industry has been talking about this notion of executable papers and papers of the future and beyond the PDF for a long time. And maybe over beer sometime we can just sort of discuss why I think these things haven't moved at the pace that they ought to have moved at. But that's sort of how I came to this talk today. It's like, talked about this for a long time, so what's really happening? Today, I work for a philanthropic organization, and we have a very modest goal of attempting to cure, prevent, and manage all human disease and suffering by the end of the century. So it's the by the end of the century part that lets us sleep at night, because we have a long time to work on this. But we also want to back up and say, you know, what are the sort of near-term goals? What are the sort of achievable, measurable things that we can do? And we, across all of our programs, attempt to set some more near-term milestones here. Within the area of curing all human disease and suffering, one of the arrows in our quiver, or one of the areas that we want to try to contribute to, is the support of new tools and technologies that help accelerate the scientific process. What are the incentives? What are the mechanisms? What are the you go back to sort of this notion of this sequential or diverging tree that Lindsay just talked about. What are the things that we can start turning science into more of an engine, more of an iterative process, more real time, building upon each other's experiences? And we believe that open science offers a lot of the sort of concepts within that. So specifically, when we start looking at the open science investments that we're making right now, we are trying to look at some of the platforms that enable sharing of scientific outputs beyond the PDF. So sharing of data sets and sharing of protocols, sharing of preprints, for example. But also today we've been talking a lot about sort of this notion of incentives and like what are the incentives? And so people who are developing research software oftentimes don't get the citations to those software, or even if they're getting citations to those software, it's not sort of recognized by their departments in the same sort of contribution to their domain as a paper in Nature or Science would be. One of the things that we're doing specifically in this area is we are running a program right now called the Essential Open Source Software for Science. In fact, we just closed last week on round 2 of an RFA, but we are making fairly modest grants to, I think in the first round, about 32 different proposals that represent an aggregate about 42 or 44 different open source software projects. And part of this is going to help them address some of the things that, you know, operating on a shoestring budget, they don't always have the time to do. So looking at issues of documentation, looking at issues of training and development, usability. They were sort of free to propose anything, but the sort of requirements were, this has to be an existing thing, not just an idea that I want to go out and build something. It has to be used by the community. So both looking at it from if it's a GitHub repo, for example, what other projects are being built upon it? How many people have started your project? How many contributors do you have? Is it a real thing? Is it an active thing? And then looking at the sort of reflection of your software project on the published literature. Are people citing it, or at least are they mentioning it in the research? And those are a couple of the dimensions that we looked at to sort of to validate that. Anyway, this is an ongoing program in that sort of recognizing people aspect of it. I'm going to sort of fast forward on from this, but one of the things I think that also makes us unusual as a philanthropic organization is that in addition to our grant making side of the house, we also have a significant technology aspect. And so the M down there in the lower right-hand corner there is a project called Meta, for example, that is a research discovery engine, current awareness engine that we're building in-house So we look at each one of these areas of, what are the things that we can complement the ecosystem, what are the things that we can invest in, what are the things that we can collaborate with the community on, and what are the things that we can, we feel like we want to build and experiment and explore with. So I know the carpentries was just mentioned. This was an investment that we made with the Moore Foundation late last year to help support the carpentries and give them a little bit more capacity to take some of these traditionally a lot of times sort of train the trainer model and in-person trainings and developing more sort of self-led online tutorials for people to address your question. How do we build that computational capacity within the biomedical community? So just a sort of a quick brain dump as I was trying to think about the what's the desiderata of the paper of the future. Many, if not most of these have been talked about today. This was my just sort of quick brain dump. Not all of these are sort of mutually consistent. Like, how can something be both open in real time, but also be, how can it be both persistent and dynamic? How can it be freely available immediately, but then also be trustworthy and sort of feel like it's gone through a peer review process? And so there may be some incompatibility in this list. But what I want to do is sort of pivot a little bit and talk about, and I guess what I left blank on the left-hand side of the slide are some of the other things that have come up today on what are the blockers, what are the obstacles to sort of achieving this. We talked about the pain in the ass aspect of data sharing, having to get it into a shape where it is shareable. What are the incentives? What are the rewards? Where does the money come from? What are the entire sort of ethical dimensions that were covered by Michelle earlier today? But the reason I changed the title of the slide from the paper of the future to the preprint of the future is one of the things that I have started to see and is developing in the preprint culture, which, to be completely honest, is a relatively new thing within the bio space, is that it offers a platform for experimentation, and it offers a platform for us to try new things, where my efforts 15 years ago with the publishing industry were sort of met, going up against an industry that has a way of doing things. They have a monetary engine that they didn't want to disrupt. Even when they were interested in experimentation, they were often sort of built on these manuscript submission pipelines that just couldn't handle new ways of looking at the data. So I'm gonna copy what Michael Keller did earlier today and ask, first of all, how many people here know what a preprint is? That's good. How many people here have ever submitted a preprint? Okay, how many people have submitted a preprint to archive.org at Cornell, bioRxiv, SSRN, another one? Any other ones? What am I missing? ChemArchive. Okay, so we have a little bit of a distribution here. So within, sorry, that was my way of figuring out what domains you were in. Within biomedicine, actually one of the first experiments in preprints happened in the early 60s. The NIH ran a program, I can't actually remember what it was called, information exchange groups, essentially a sort of a mailing list for preprints. And it only ran for a few years. They had a few thousand preprints that were being sort of circulated at the time, but not unlike what we've seen in the past few years. Publishers didn't really like it. They sort of objected. And so within the biomedicine community, they sort of retreated from that. It did manage to take hold, starting with, again, it was a paper routing system at Los Alamos Labs. It then evolved into an FTP server, and then later moved to Cornell University, archive.org. High energy physics expanding into other areas of physics. They also have math and computer science and a little tiny bit of bio in there. But then in the past couple of years within biomedicine, bioRxiv has started up at Cold Spring Harbor. And in fact, they're built on top of Highwire Press, which came out of Stanford here. And an interesting, people haven't used bioRxiv before, this is what it looks like. You can download both a PDF version of the submitted preprint, but they also do, within 2 or 3 days, they do an XML conversion of the preprint. So if anybody wants to get full text access to XML versions of the preprint, those are all easily available via their APIs. The growth within the bio space over the past, whatever that is, 13 years, has really sort of taken off, with the green portion of the graph here being the bioRxiv portion of it. So you can see sort of some of the other players. The red is the QBio channel within Archive at Cornell. Nature Proceedings died off a few years ago. Faculty of 1,000 Research is in there, and they just got acquired by Wiley, I think, Taylor and Francis, I can't remember, in the past couple of weeks. So this has really sort of been, you know, a great thing for bioRxiv, I think a great thing for the biomedical space. What I don't have on here is a graph of what percentage of the actual publications that represents within biomedicine. And the answer is it's still about 3%. So while it is having this exponential growth relative to all of the research in bio, it's still extremely modest. But we were also starting to see people using the preprint server at bioRxiv for a number of things, a number of use cases that you don't often see getting published. Negative results, contradictory results papers, data papers, a number of other things. And we started the conversation with Richard Siever and John Ingalls at bioRxiv about sort of looking at this as a catalyst, perhaps, for running some new experiments. How can you take some of the outputs that we're seeing out of the preprint ecosystem and start feeding those things back into inputs? And we just, about 2 months ago, in the middle of December, got a group of people together up in San Francisco from the biomedical community. Journal of Open Source Software was just mentioned by Lindsay earlier. Arfin Smith was there. A number of different publishers and preprint repositories. And we sort of spent 2.5, 3 days starting to sort of brainstorm what we wanted to sort of see develop from a protocol perspective, a tooling perspective, an integration perspective. And I want to give just sort of a couple of examples that came out of this conversation, or examples that were sort of brought up during the course of the day. These are not things that Chan-Zuckerberg has funded, but just to sort of seed the idea of using platforms like preprints as a way of experimenting with new types of scholarship and new types of peer review, perhaps. So one of them was a project called JetFighter, which, when you're dealing in an open access world, when you're dealing with the way that the content can be accessed via APIs, and specifically with bioRxiv, since they allow versioning of the preprints, it opens up this possibility of having a programmatic conversation with the author of the manuscript. And so what the people who wrote JetFighter started to do was to run the images in the preprints through a series of accessibility tests. And then if they failed those tests, automatically pinging the authors saying, hey, if you use a different color palette, your images will be more accessible. And by and large, the responses that they got from the author was, that's a really good idea. I'm going to do those. They would update the images. They would resubmit into the ecosystem. These may or may not be things that would also be caught as part of a peer review or editorial process on manuscript submission, but this doesn't gate the availability of the research getting out and getting discovered. And for a significant portion of the population, it probably doesn't even affect their ability to take in the scientific findings from the paper. But it makes the paper better, it improves it over time. We also had another participant who was from Argentina and was lamenting the fact that, by and large, the scholarly conversation is going on in the English language. And he was working with a lot of researchers who were using bioRxiv in Argentina, where English was not their first language. And over the course of the first day and a half of the conference, 2 or 3 people got together in the corner of the room and started working with some tools like Google Translate, and made an entirely new on-ramp that sort of routes the entire portal of bioRxiv through Google Translate. And since the full text of the papers are also available in HTML on the site, you can go not just from the browsability and search results, but down into the full text of the paper and read them in non-English language, which is what they're primarily published in. This starts opening up other possibilities, and there are the abilities right now of creating. One of the problems with tools like Google Translate is that they don't understand always the domain-specific language, and there are processes or services now that you can actually feed data dictionaries into a machine translation model, and you sort of can imagine having a specific biomedicine machine translation model, which is something that we could improve on this. Another example that Michael Keller started to refer to this, or a related project out of Springer Nature earlier today. Springer Nature and their Biomed Central open access press, in partnership with an organization called Preprint hosting service called Research Square, has just started up something called In Review. I believe it's only rolled out right now to their open access publications in Biomed Central, but according to Salmia at Springer Nature, they do eventually plan to roll this out to all Springer Nature titles. And what it does is it integrates the preprint deposition process with manuscript submission. So if an author goes to a Springer Nature journal and says, here's my manuscript for peer review, they're given the option of simultaneously having a copy of that manuscript deposited as an open access preprint on this preprint repository. A. B, the peer review process happens within that preprint repository, both from a public access and commenting on the publication, but then also the editorial staff of the Springer Nature journals are putting their public comments on that. The versioning of that, if they are asking the author to revise part of it, that versioning then happens on the preprint repository. And at the end of this, you're either going to get a decision to publish that in the Springer Nature Journal or a rejection. And in the case where they publish it, then this entire history of the editorial process, sort of a form of open peer review, is preserved in the preprint repository. It's sort of snapped to the most recent version of it. And then as well, there's a version that gets, sort of version of record, if you will, gets published in the Springer Nature title. If the preprint is rejected, they roll back all of the changes in the commentary that happened on the preprint server, so back to the originally submitted version, and they just sort of walk away. But in either case, you get the advantage of sort of shortening the availability of that research to the research community. And I think, getting back to this notion that Lindsay talked about having an engine of science and sort of moving away from what I see as one of the issues of having this entirely serialized process of I do my research, I publish my research, I wait till it goes through peer review, then it gets seen by the world and read and built upon, into something that's much more interactive and versioned and visible and transparent, I think is a positive step and a way that preprints are sort of challenging or enabling some of these notions of open peer review. I think there's also a pilot going on right now, as a matter of fact, between PLOS, maybe just PLOS One, but PLOS and bioRxiv is doing something similar here. And then another example, are people aware of sort of the notion of pre-registration? So generally pre-registration in the biosciences is saying, this is the experiment that I'm going to run. This is the protocol that I'm going to use. The Center for Open Science, another preprint hosting service and other things, in partnership with PLOS is running something right now called registered reports, which takes this notion of pre-registration and puts it into the sort of equation where you take your protocol, you take your proposal, your design study, and you send that in for peer review. Before you've ever run any experiments at all, you say, this is the research that I'm going to do. And it allows you to run that through peer review, where your statistical methods might be questioned or your number of study participants might be questioned. But you revise that and you get pre-approval, pre-registration to have that published before you've even ever conducted your experiments. So now that's out there. That's a published registered report. And you have the guarantee at the end of your experiment of also having your experimental results published, even if it's negative results. So you have sort of these two artifacts. One is the study plan, and the other is the study results that go out there. So they're running a pilot of this with PLOS, and again, not preprints per se, but sort of in that sort of same vein of more transparency, more availability of negative results, and hopefully more acceleration of the scientific process. I was going to talk about Jupyter Notebooks, but I don't think I'll talk about that now. I think they're great, though. It's awesome. Other than to say with Jupyter Notebooks, I mean, I think some of the questions that were being asked, you know, from a library, from a preservation point of view, from an identification point of view, certainly some great questions that come out of that. Like, what are these reproducible artifacts? How are they identifiable? You know, other than to say, in many ways, and Lindsay alluded to this, you know, the software development world has, to a certain extent, solved some of these problems. Software is constantly evolving. It's shifting. Michael this morning talked about that. You know, research papers are static, and software is this constantly evolving thing. And I'm trying to come up with the argument for why we shouldn't be viewing research as a constantly evolving thing. We do have this sort of problem of identification, and you know, a DOI represents a paper. Are there corollaries in, and there are some examples with the Zenodo repository, for example, where you can actually get a DOI issued for a snapshot of your source code. I'm not entirely sure that that's like a fully sustainable model over time, but you know, we should be able to refer to a version of the code or a timestamp of the code in this sort of evolving source code model. But ultimately, we started to identify some of the areas, some of the integration pain points. Preprints today are built on a lot of different platforms. There are not necessarily consistent file formats. I mentioned that bioRxiv right now is converting into JATS XML. Archive.org, as far as I know, doesn't have a JATS XML version. They have you know, either PDF or maybe they have their own proprietary XML or localized XML. But anyway, software licensing is another big problem. People tend to have this sort of mental equation of preprint equals open access. The reality of the situation is that licenses are woefully inconsistent. To take bioRxiv as an example, when you submit a preprint to bioRxiv, you're given the option of what license to choose. Most people don't know the ramifications of the license choice. And then you sort of have this sort of, you know, random generation. So one of the things that we're starting to think about as a funding agency is starting to come up with, along with the other funding agencies, recommended licenses that we can then feed into the manuscript submission workflow for bioRxiv and in the publishers. So if you come in and say, this is my research, and I did it with funding from the Chan-Zuckerberg Initiative or the Wellcome Trust or HHMI or Gates Foundation, in the back end, they can say, would you like to use a preferred license from your funding agency? And sort of have that go through without it needing to be an end user choice. All right, let's get really immediate here for a second. Just like one last sort of stab on why I think preprints offers us a worthwhile way forward. NCOV, novel coronavirus, actually just got a new name today. The CDC gave it a new name. Franklin asked me to submit my slides last Friday. This is a dashboard that Johns Hopkins University created last week. So real-time aggregation of data sets from all over the world talking about the spread of the novel coronavirus. It's zoomed in here on China, but you can pan around any part of the globe. I just took another screenshot this morning. It's up to 43,000 confirmed cases. The level of the level of information sharing that's gone on over the past 6 weeks around the coronavirus has been astounding. In many cases, comparing back to SARS, the Chinese government has been a lot more forthcoming with sharing data this time than they had been previously. But there's still some issues with doing even data aggregations like this, which is, even though we're getting data in around the cases of coronavirus, it's a little bit unclear about what the dates are assigned with it. Is the date the first onset of symptoms? Is it the date of diagnosis? Is it the date of hospitalization? So trying to come up with the sort of models is limited by how good that metadata is, and the ability to aggregate it is also limited by the same factor. But there was a study that was done in PLOS Medicine a couple of years ago, looking at a couple of previous outbreaks, Ebola and Zika, and looking at the median time saved by publishing preprints as opposed to waiting until the published version of a paper, peer-reviewed published version of the paper came out. So you have here sort of along the x-axis time, you can see when the World Health Organization had declared an emergency declaration on it. When Zika came out, there's actually a shorter time window between a global health, things should be made available as quickly as possible as a preprint. But I think in the case of even with Zika, there was basically a median 150 days delta between when papers that were available, research that had been made available via preprint versus research that was made available as a post-publication. So if you start like now overlaying that with what we've seen over the past month with coronavirus, really starting about the 1st of the year, 31st of December, after a week, globally, the scientists, and in fact, this was like one lab who was talking about with the data that they were given, they had identified, sequenced a related virus that existed in bats, created a diagnostic test as serology, isolated the virus, and then created some experimental validations over the course of a week. Compare that with the 150-day median previously. So it's shining a light when we're sort of in the middle of a global outbreak right now on some of the validity or some of the imperatives, I would say, around rapid research. This has raised a question, and there was a number of reports that were coming out over the past couple of weeks around the issue of the quality of the research that was coming out. One person tweeted the fact, and this was a week ago Friday, I think, I don't have the dates on here, but a week ago Friday, they said, hey, there's 313 preprints on bioRxiv right now. And it's sort of a call to arms to the global research community to dive into those 313 reports. And over the course of the weekend, almost every single one of those had positive or negative feedback on the research that was being done. And out of that sort of came one report that in some ways could be interpreted as sort of feeding some of the conspiracy theories that were going on. The people who published the report said, I think you guys are misrepresenting what we're saying, but we don't want to spread this misinformation, and they took the report down. So this was from Friday, identifying 313 preprints that needed review. One of them that was causing a lot of consternation amongst the global research community. By Sunday afternoon, the research team had decided to take it down. And you sort of think about that window of roughly 48 hours versus what happens in the retraction world today. How long does it take between the time when a paper that gets peer-reviewed and gets published, if it's been identified, that retraction can often take 12 to 18 months before that paper is actually taken down. So I see this as a positive step in community peer review, in sort of proper peer review that can happen and enabled by A, the versioning, and B, the sort of quick response to this. And then the last thing I will mention is not just the paper as the narrative, the PDF version of this is the experiment that we ran and this is what we found, but like data and like software, starting to decouple some of the other elements like the methods and protocols from papers. So protocols.io, run by a small shop out of Berkeley here, is a publishing platform for protocols not needed when you're sort of really thinking about this in sort of a, a computational environment like a Jupyter Notebook. But if you're talking about a wet lab environment where you're talking about a set of procedures that you're going through in your lab, protocols.io allows you to submit this as a machine readable, re-executable workflow. And you can actually run multiple runs of that workflow within the tool. So there's a couple of examples here for both sequencing and detection protocols that were developed and submitted into protocols.io concerning the coronavirus over the past week. So fragmenting the literature or decomposing the literature into data and software and protocols, accelerating the pace of the discovery and the versioning of it is all, I think, characteristics of the paper of the future. And the question is, why do we need a virus to sort of cause us to realize this? Why can't this be a part of how science gets done all the time? So with that, thank you.